actually want to introduce myself, which I always forget to do. My name is Mark Rosenzweig, and from West Bloomfield, Michigan, and 11 years into my journey. And I want to welcome everyone to an ALK Talk and welcome back Dr. Shaw, who's been here 3 times and who's supported us and many times during the summit. So really appreciate that. And I want to go ahead and start up some polling questions.
Here we go. First question, when were you diagnosed? Happy New Year, everybody. Thank you so much for joining us this Sunday evening with Dr. Shaw. We are so happy to have you here. I'm Summer Farman, by the way, diagnosed in 2020 and recovering from influenza A. So get your flu shot, everybody, because I was a little delayed and I'm paying the price.
Oh. Okay, I'm gonna end the poll and share the results in a second.
Okay, here we go. When were you diagnosed? 13% of you were under a year. So this will be a real treat for you getting to meet Dr. Shaw. 9% of you are 1 to 2 years. 22% of you are 2 to 3 years. 16% of you are 3 to 4 years. 13% of you were diagnosed 4 to 5 years ago. 10% of you were diagnosed 5 to 6 years ago. 8% 6 to 7. 3% 7 to 8 years. 3% of you 8 to 9 years. Oh, this is awesome. And 5% of you were diagnosed 10 years or more. Okay, and what type of treatment are you receiving? We will be having in February treatment roundtables on a Sunday evening during ALK Talk time. This is a great time to join for whatever your TKI is, or if you're in a clinical trial, or if you are on chemo and a TKI, and you can talk to other people on the same treatment as you. We did that last year, and it was a big hit, very fun, a little chaotic. But so look for that coming up next month or later this month, possibly.
Hey, Summer, do you want to take it away, or is your voice not doing it?
I got it. I got it.
All
Can you?
right, all right.
This is it, though. Okay, what type of treatment are you receiving? 5% of you are on chemo and a TKI. 50% of you are on alectinib. 9% of you, plus me, are on brigatinib. 27% of you are lorlatinib. 1% of you are straight chemo. 5% of you are on a clinical trial. 1% of you are no treatment, and 1% of you are not applicable. Thank you for participating, and thank you for joining us.
I want to introduce our guest, Dr. Shaw. I met in 2014. She was speaking at a conference with Dr. Kamich, and she's been in the ALK space since 2007. So I want to introduce Dr. Al Shaw. Dr. Shaw, take it away.
Great. Well, thanks so much, Mark, and hi, everyone. It's really great to be back and to see some familiar faces and names in the audience. Hi, Emily. I think the last time I spoke, actually, it was at the ALK Summit in the summer of 2023, I believe. So it was about 18 months ago. And at that time, I was still at Novartis leading early clinical development in oncology. But I think some of you know that 6 months ago, I came back to academia to serve as the inaugural chief of strategic partnerships at Dana-Farber and to resume research in patient care and thoracic oncology at Dana-Farber. So I'm really thrilled to be back. So the first thing I'd like to do is share my slides. I decided that it wouldn't just be all Q&A, that maybe we'd start with just a little bit of background, if that's okay with everyone. So give me a moment to share. And let me know, Summer or Mark, can you see these slides?
Yep, great.
Okay, great. All right, well, here we are. So I titled just the introduction here to my sort of talk as ALK positive lung cancer in 2025. And I really want this to be, reflecting on 2024, but also really forward looking as well. And so, I thought I'd spend the first 10 minutes or so reviewing some of the highlights from 2024 that are directly relevant to ALK and to many of you. And then I thought I'd spend maybe another 10 minutes or so on some emerging and I think exciting new approaches for ALK positive lung cancer. And just as I did at the ALK Summit 18 months ago, I'm hoping to focus this section on what we often refer to as sort of orthogonal approaches. So not just targeting ALK and inhibiting it, since we already have such effective ALK targeted therapies, but actually going beyond ALK TKIs and developing alternative and completely different ways to target an ALK positive lung cancer. And then we should have at least 30 minutes to have an open discussion and Q&A. Ken, I think you're the one who called this Ask Alice session. I think there should be plenty of opportunity to talk about whatever may be top of mind for you guys. All right, so with that, I'm going to turn to the highlights in the field from 2024. And I think since I heard in that polling question that some of you probably are fairly new to the group and perhaps more recently diagnosed with ALK positive lung cancer. So I thought I'd actually start with just a little historical context to help frame the two highlights that I plan to mention. And so this slide captures I think how much the field of ALK positive lung cancer has really changed over the last decade. Some of you may remember that about 10 years ago, when I met Mark, actually, the standard treatment for patients with advanced ALK positive lung cancer consisted of the first generation TKI, crizotinib, followed by a more potent second generation TKI, such as ceritinib, alectinib, or brigatinib. But this shifted almost overnight, actually in 2017, when we presented the results of the global ALK study at ASCO and showed that alectinib was significantly superior to crizotinib and more than doubled the progression-free survival. At that same time in 2017, we also presented compelling data on a third generation ALK inhibitor, lorlatinib, from the phase 1 study. And this is even more potent and CNS penetrant than the second generation drugs. And so the field pivoted from sequential first and second generation TKIs to sequential second and third generation TKIs, as I've shown here. Now, over the next 5 years or so, we continued to use primarily alectinib, some brigatinib first line, followed by lorlatinib second line, although we did already start to generate data on lorlatinib suggesting really remarkable activity in the first line setting. But I would say in this time period, the major advance was the development of yet a fourth generation ALK inhibitor, NVL-655, which had been specifically designed to overcome all of the known resistance mutations in ALK, had been designed like lorlatinib to be CNS penetrant, and then lastly, also designed to not inhibit other related proteins such as TRAC, which can cause side effects. And so in 2023, many of our ALK patients were now receiving sequential second, third, and fourth generation TKIs and deriving durable clinical benefit from each one. And so the two major highlights from 2024 that I wanted to mention tonight include, of course, the longer follow-up data with lorlatinib that now pretty firmly positions lorlatinib as the preferred first-line treatment for patients who are newly diagnosed with ALK-positive lung cancer, and also additional supportive data with NVL-655, really validating its activity and also its really favorable safety profile in our ALK-positive patients. And so let's start quickly with lorlatinib first. And I know many of you are familiar with this. This is the CROWN study, so a phase 3 study. We published it back in 2020. And just as a reminder, these were all newly diagnosed patients. They had stage 3B or 4 ALK-positive lung cancer. And they were randomized to receive either first-line lorlatinib or first-line crizotinib. And some of you may ask, why did we randomize to crizotinib at that time and not alectinib? Well, at the time that we launched the study, we actually didn't have the results back yet from the global ALK study. And crizotinib was actually still considered the first-line standard of care. And so here are the results of the CROWN study after a median follow-up of 18 months on the left and then 3 years on the right. And you can see, I think, in both graphs that there's large separation between these two PFS curves. Lorlatinib easily beat crizotinib in terms of progression-free survival. At both of these time points, actually, we didn't reach a median PFS with lorlatinib. And actually, when you compare to crizotinib, lorlatinib was able to reduce the risk of progression or death by about 73%. So really significant. Now, despite these results, there wasn't actually that much uptake of lorlatinib as first-line therapy. I think alectinib was still very much favored because of the ALK's data. We knew that median PFS with a frontline alectinib was in the range of 3 years. And of course, alectinib, by and large, is really well tolerated. But again, this changed almost overnight in 2024, just at ASCO this year or last year, when Ben Solomon presented now the 5-year follow-up of the CROWN data. And here again, you can see this marked separation in the progression-free survival curves. And I think what was remarkable and really practice-changing at that time is that you can see there's no median progression-free survival still reached in over 1/2. So about 60% of patients at 5 years still had no signs of progression. And so at that time point, lorlatinib had further reduced even the risk of progression or death compared to crizotinib by over 80%. And I think what was also really striking to many of us in the field were the results in terms of the brain or CNS activity. And as you can see here, these are, again, curves related to intracranial progression. And you can see lorlatinib was able to protect the brain. So 92% of patients who received lorlatinib had no signs of CNS metastases after a median follow-up of 5 years. And actually, for those patients who didn't even have brain metastases at baseline, this rate was even higher at 96%. So I think pretty amazing activity in the brain, also protection of the brain by lorlatinib. Now, I included this slide here on the safety profile of lorlatinib. There were no new safety findings at this longer follow-up, but I think those of you who are on lorlatinib now appreciate that this TKI can have some challenges, especially compared to other TKIs. And we can discuss, more of these side effects in detail during the Q&A, but just to mention that some of the side effects, they do require active management and often dose reduction. These include, as shown here in this graph, fluid retention and edema, elevated cholesterol and triglycerides, increased appetite and weight gain, neurocognitive and mood side effects in some patients. And again, we can come back to these in the Q&A, but I did want to just emphasize that these side effects are reversible. They're very manageable, and dose reduction can be really effective in mitigating the side effects without compromising the efficacy. So now moving to the second major highlight from 2024, I wanted to spend just a few minutes on the 4th generation ALK inhibitor NVL-655. I already mentioned that 655 was rationally designed to overcome many of the limitations of prior ALK TKIs. So it should cover not just single ALK resistance mutations, but also compound ALK resistance mutations. It was designed to be highly selective to avoid hitting this other protein called TRAC, which may be mediating some of the side effects of lorlatinib. And it's also really CNS penetrant. So it should be able to treat and protect the CNS. And so here's the latest data from the phase 1 study of NVL-655. This was presented by Alex Drillon at ESMO in October. And in the waterfall plots, you can get a sense that this drug is quite active across a significant fraction of ALK patients. If we focus on just one of the subsets of patients, those who had received 2 or more ALK inhibitors, including lorlatinib, you can see that the response rate's about 35%, but it's even higher, coming in at around 50 to 64% in those patients who actually have a documented ALK resistance mutation or a compound mutation. So remember, this is phase 1. So this is really compelling activity, even in patients who've already received a number of prior ALK inhibitors. Now, I think what's even more compelling with 655 is its safety profile. And so shown here are the most common side effects that we've seen in the phase 1 study. As you can see, ALT and AST refer to liver function tests, and elevation of these are the most common side effects occurring in about 1/3 of patients or so. Most of the time, these are just mild elevations, really not requiring any intervention, although in about maybe 10 to 15% of patients, the liver test abnormalities can be high enough to require a dose hold and even a reduction. There are a few other side effects that you can see here, like constipation, change in taste, maybe some mild nausea, but they're just not that common and they're almost always mild. So really what you're seeing is a really well-tolerated drug with really none of the most problematic side effects that we sometimes see with lorlatinib. And so based on these really promising phase 1 results, NVL-655 is moving very rapidly in the clinic. We're currently enrolling patients into the phase 2 portion of the study across 6 different cohorts, as you can see here. And I believe actually across the entire study, there are now almost 600 patients who've been treated. So we're gathering lots of experience. We're also just about to launch the phase 3 study of NVL-655 in patients with newly diagnosed ALK positive lung cancer. This is going to be the ALKazar study. And then here you can see that we're comparing NVL-655 to alectinib, and I'm sure there'll be questions around that study design, and we can talk about that during the Q&A. Okay, so with those highlights from 2024, let's turn to 2025 and beyond and talk about what I think are some exciting new approaches to tackling ALK positive lung cancer. And again, all of these really are going to go beyond just ALK TKIs. And so the first approach I wanted to highlight is a potential new target in ALK positive lung cancer. This target is called MTAP, and this is an enzyme that acts as a tumor suppressor by regulating the methionine salvage pathway and a number of other cellular processes. And if you lose both copies, you can lose both copies of this MTAP gene. We also call that homozygous loss or homozygous deletion of MTAP. And we see that in about 10 to 15% of all cancer. So it's not just lung cancer, it's not just ALK positive lung cancer. And then notably, we often see it together with loss of another tumor suppressor called CDKN2A. Now, in this graph in the middle, you can see that the frequency of MTAP deletion varies by tumor type. The highest frequencies are seen in primary brain tumors, mesotheliomas, esophageal bladder, pancreatic cancers. But we do see MTAP deletion in other cancer types like non-small cell lung cancers. The frequency of MTAP loss is about 10% overall. Now, why is MTAP loss important? Why am I telling you guys about this on a Sunday evening? Well, without going into the details of the biology, suffice it to say that when a cancer cell has lost both copies of this MTAP gene, that cancer now becomes particularly sensitive to inhibition of another enzyme. This is an epigenetic regulator called PRMT5. Now, multiple preclinical studies have shown that cancer cells with MTAP loss are more vulnerable to PRMT5 inhibitors than normal cells. And that's then leads us to basically refer to PRMT5 as a synthetic lethal target. In other words, inhibition of PRMT5 is only lethal in the setting of the MTAP deletion in cancer cells. Now, there are multiple PRMT5 inhibitors that have entered clinical development in just the last few years, including ones from Amgen and Mirati, BMS, and Tango. And we're just now starting to see the clinical data emerge with these new targeted therapies. This is phase 1 data from one of the PRMT5 inhibitors. And as you can see in this waterfall plot, there's good activity here with this PRMT5 inhibitor across different tumor types with this MTAP loss. And that includes responses in about 1/3 of lung cancers with MTAP loss. We still, follow-up is still ongoing. This is phase 1 data. We don't know how long these responses will last, but so far the average duration of response exceeds about 6 months, which is really encouraging. And we've seen other very comparable data from other companies with their PRMT5 inhibitor. So I do think this is now a clinically validated target in a molecularly defined subset of patients. And so why is this relevant to you guys, to ALK? Well, since I arrived at the Farber 6 months ago, I have been really interested in studying this new target in lung cancer. And I've been working with one of our fellows from Gustave Roussy, Miha Aldea. And we've now looked for MTAP deletion across all the different oncogene-driven subsets of lung cancer. And interestingly, what we found so far is that among these different subsets, ALK, which is shown here on the far left, has the highest frequency of harboring MTAP loss, with about 30% of our ALK cases showing homozygous loss, so loss of both copies of this MTAP gene. We then teamed up with Marie-Anise Ayouaket from Pasiani's lab to look at the impact of losing MTAP in the setting of ALK. Of course, these are all laboratory-based experiments. And just to share with you, on the left, there's an ALK positive cell line that either has intact MTAP or has been engineered to lose MTAP. And actually, those two cell lines have nearly identical sensitivity to alectinib, actually, or lorlatinib in vitro. So loss of MTAP probably doesn't impact at least the initial sensitivity to ALK inhibitors. And then on the right is preliminary data with an ALK positive cell line with MTAP loss, where we're treating the cell line with either lorlatinib, which is shown in orange, with the PRMT5 inhibitor in green, or with the combination of ALK and PRMT5 inhibitors, and that's in red. And I think you can appreciate that with either lorlatinib or this MRTX drug by itself, there's some activity in this in vitro assay, but the combination, that line, that red line you see, indicates that there's much greater activity with the combination. And that suggests that there's at least additive activity with this combination, and that patients, the 30% or so of ALK patients who have MTAP loss, a combination could actually induce even deeper and more durable responses than an ALK inhibitor by itself. And so to begin to understand whether or not these findings are clinically relevant, how we could even think about advancing a combination like this to the clinic, we've been screening all of our ALK patients for MTAP loss and starting to enroll patients onto our clinical trials of PRMT5 inhibitors. And I wanted to share just one illustrative case with you. And this is the first ALK patient who I recently enrolled onto a clinical trial of a PRMT5 inhibitor. So this patient is 75. He was diagnosed about 2 years ago, almost 2 years ago, with advanced ALK positive lung cancer. He had metastases to the lung, bone, and brain at diagnosis. Initially, his testing was done at Foundation Medicine. They detected the ALK rearrangement, also a number of other genetic alterations, including MTAP loss. He was then started on first-line alectinib, and he did well initially, but after just about 8 months, scans suggested early progression, and he ultimately had to come off alectinib after about 11 months because of ongoing progression. And of course, that's a lot shorter than what we would expect with first-line alectinib. He underwent a repeat biopsy, and that showed a similar profile as before, and notably no evidence of ALK resistance mutations. He still then went on to second-line lorlatinib, and unfortunately, he didn't respond at all. In fact, he developed now symptomatic progression. And at this point, he was transitioned to standard of care chemotherapy, and he went through 4 cycles of chemotherapy, but scans showed worsening disease, and now his symptoms were really escalating. And so at this point, he was referred to me. And given that he had MTAP deletion noted in his biopsy, I actually went ahead and we enrolled him on a clinical trial of a PRMT5 inhibitor. And for the first week or two, I was pretty concerned because his disease-related symptoms were actually even worsening a bit. But then after about 2 weeks, his symptoms started to ease up. And by about 6 weeks after starting this PRMT inhibitor by itself, all of his symptoms have resolved, and he's actually been tolerating this new drug incredibly well with almost no side effects. And so shown here are this patient's baseline and restaging scans after just 6 weeks of treatment. On the left, you can see 2 images of his lungs prior to starting on the PRMT5 inhibitor. You can see there's a very large mass in the left lung and multiple lung metastases, other disease, which you can't see here on the scan. And then if you look on the right, I think you can appreciate those are his first scans after the 6 weeks of this drug. And I think you can appreciate there's been a really dramatic response in his lungs. And that's, of course, consistent with the resolution of his disease-related symptoms. So really a remarkable response to this new targeted therapy. And just remember, this is a TKI-resistant patient. He's already received 2 of our best ALK-targeted therapies. He's received chemotherapy and progressed, and now he's responding to just this PRMT5 inhibitor by itself. And so based on the preclinical data and now some emerging clinical data, including this case and other cases that we have of oncogene-driven lung cancers responding to PRMT5 inhibitors, we're hoping to move quickly and try to establish the role of PRMT5 inhibitors, this new class of targeted therapies in ALK-positive lung cancers that have MTAP loss. And last month, we did submit a proposal to actually the ALK Positive group to consider supporting further investigations into MTAP and PRMT5 inhibitors. And here are some of the questions that I hope we can address, including further preclinical evaluations of the combinations of ALK and PRMT5 inhibitors, both in the resistant and the TKI sensitive setting, and really trying to figure out if there's true synergy or additivity between these two classes of targeted therapies. Also looking preclinically at whether or not if we use these two targeted therapies in combination up front, if that could delay or maybe even completely prevent the emergence of resistance. And then, of course, if the preclinical studies I think are promising, I would love to test a combination of ALK and PRMT5 inhibitors in the clinic in an investigator-initiated trial for ALK patients who've become resistant to ALK TKIs, as well as for ALK patients who may still be TKI sensitive. Okay, in the last few minutes, I want to turn to just another key approach that we and many others are pursuing in the field, and that is to target the minimal residual disease state, which we call that the MRD state, and we know that's comprised of these so-called drug-tolerant persisters. And I think some of you may be familiar with this concept. It is the focus of our breakthrough cancer, lung cancer team lab. But just to remind everyone, this is a figure from an ALK review that we published about a year ago. While ALK TKIs are highly effective and can eradicate a significant proportion of ALK positive cancer cells, they really can't eliminate every last cancer cell. And in fact, there is a small subpopulation of cancer cells that manage to survive the ALK TKI and persist. And the exact mechanisms that enable these so-called drug-tolerant persisters to survive are really not known. It's almost certainly not genetic mechanisms, but however they manage to survive, these then provide a nidus for the eventual emergence of full-fledged resistant cancer cells. And so for the last 5 years or so, a lot of our research has shifted to focusing on how to characterize these drug-tolerant persisters and, importantly, how to eradicate them with the goal of then completely preventing the development of resistance. Shown here on the right are examples of potential strategies that we're taking or considering to address drug-tolerant persisters. This includes local radiation therapy to residual disease. Adding chemotherapy is another approach to try to minimize residual disease. We're also really interested in adding another type of targeted therapy. These are called YAP-TEAD inhibitors because we know that YAP-TEAD signaling may be important for the survival of drug-tolerant persisters. And finally, many here, I think, are very familiar with efforts to harness the immune system, primarily through an ALK vaccine to eradicate residual disease. That's, of course, a project led by my colleagues, Roberto Chiarla and Mark Awad. But in this last slide, I just wanted to share some very new data from the labs of Pasiani, Dave Barbie, and Eric Smith at Dana-Farber. This was presented already at the World Lung Conference that some of you may have attended in September. And just to note, this is focused on EGFR mutant lung cancer, but I think the principles and the learnings are likely going to be similar for ALK positive lung cancer. And so briefly, Simon Baldacci, who is a postdoc in the lab, was interested in how to target drug-tolerant persisters in EGFR mutant lung cancer. And he analyzed persisters that had been generated in the lab, so in vitro, as well as in a patient who received neoadjuvant EGFR TKI, and then looked at the differential protein expression in persisters versus sort of baseline specimens. And interestingly, they found that this protein called TROP2 was significantly enriched in the persister population. Now, some of you may have heard of TROP2. It's really a hot cell surface target for antibody drug conjugates. That's another class of therapies out there for lung and other cancers. In fact, there are several TROP2 antibody drug conjugates being actively developed and used in lung, breast, expressed in other cancers. And so Simon decided to test the activity of a TROP2 antibody drug conjugate against these drug-tolerant persisters. And he found that, kind of surprisingly, the antibody drug conjugate had very modest activity. So they then turned to a different type of therapy. They turned to a homegrown TROP2 CAR T. And just to remind you guys, CAR Ts, these are T cells that have been genetically engineered to attack TROP2 expressing cancer cells. And now when they use the CAR T, they actually saw really significant tumor activity as well as improved survival in these preclinical models. And so now we're looking very carefully at persisters and TROP2 expression in other models, including ALK positive lung models. And I think if we just step back even further from this, I think we all need to be really open-minded and consider not just sort of the established modalities like chemotherapy and traditional radiation therapy, but really be open to more innovative modalities such as CAR Ts, other cellular therapies, radioligand therapies. I spoke about a lot of these at the ALK Summit in 2023. And just to really emphasize that there's incredible innovation going on, especially on the industry side. And my hope is that now that I'm back on the academic side, I can leverage some of that innovation to advance new therapies specifically for ALK positive lung cancer. And so with that, I'm going to pause here. That was a lot in about 30 minutes, but I'm happy to open it up to questions.
First of all, that was a lot more information than I expected. So,
Yeah.
and Ken,
Not
I
at
need
all,
you to
Mark.
really dumb this down for us because this Ask the Alice segment, I didn't expect to see all this new stuff, which is exciting. So, can
Well, I
you
wanted to start the
elaborate a
new
little
year
bit?
off in a good way, Mark, right? Just
Yeah, for sure. And
You
if you can
did.
stop sharing your screen, we can see
yeah, I
you
will.
bigger.
Alice, thank
So,
you.
Ken, I'm going to go to you first.
Can you hear me, Mark?
I can hear you.
Okay, great. So I think it would be worth everybody knowing, that Alice isn't just a great clinician, researcher, and communicator. If you speak to the leadership at Nuvalent, they'll tell you one of the reasons why they developed NVL 655 is because of Alice. That Alice had the ideas, and Alice was a major inspiration. So we have lots of reasons to be thankful for Alice. So thank you, Alice.
Yeah. Well, that team is
Let's
amazing at Nuvalent. So they've done incredible work. Really excited for their molecules.
So are we. And sticking with the NVL 655 for a minute, there were a number of questions in there about ALKAZAR, the phase 3 trial, and why versus alectinib. Is there a crossover? Why would anybody want to go on that instead of alatinib based on the CROWN data that you showed? Can you speak a little more about it?
Yeah, it's a complex topic. Honestly, it's not that dissimilar from the CROWN study itself, which, as I noted, in the CROWN study, we compared first-line lorlatinib with first-line crizotinib, and but I mentioned that for CROWN, it happened just before alectinib's first-line data was sort of released, and so we were able to kind of squeeze it in. And the reason that in the CROWN study, crizotinib was sort of favored as well was because, of course, we knew that crizotinib would have a much shorter progression-free survival, hence the bar would be lower. I would say it's somewhat similar in the ALKAZAR situation that Nuvalent's been planning this phase 3 trial for some time. And I think, as I mentioned before, the 5-year update of the CROWN study was presented and published in June of this year. There wasn't very much uptake of lorlatinib. I would say both because it wasn't known how long the durations of response to first-line lorlatinib would be. And as we've talked briefly about already, and some of you probably are experiencing, there are quite a few side effects with lorlatinib. And oftentimes, I would say the vast majority of patients will require dose reductions of lorlatinib. So I think the tolerability, there's a tolerability issue as well. Everything changed, as I mentioned, with the 5-year update, because now we did have long enough follow-up, 5 years, to say that, in fact, there's still not a sort of a median time. And as I showed in the slide, 60% of patients, even at a median follow-up of 5 years, were still progression-free. So we don't have a median. So we don't even have a bar to beat with lorlatinib at this point. There will be at some point a bar to beat. The median may come in, Ken, I don't know, in the 6 year, 7 year, we really don't know. So you can imagine, Ken, because you were at a company as well, it's very hard to design a study against a comparator where you don't know what that level is. But I think there was also a timing thing. This phase 3 study was planned, was underway and planned in discussion with regulatory agencies as well. And there was general overall agreement that at the time the study was planned, that alectinib was an appropriate first-line comparator. Now, in this country where we very much, of course, I think embrace lorlatinib now as the first-line therapy based on the 5-year CROWN data, I do think it will be challenging to enroll patients to that study. Having said that, I've been a little bit surprised over the last few months that I've been in clinic now that quite a few patients that we've seen have been offered lorlatinib first line and actually have chosen alectinib, again, because of the concern around side effects. And knowing that they can still go on to lorlatinib, they could still go on to Nuvalent, they have other options. And so, Ken, I think in this country, yes, it may be a hard sell to enroll patients. I recommend first-line lorlatinib, for example, but I may have patients who actually want to have a shot at Nuvalent as a first-line therapy or also are very comfortable proceeding with alectinib as their first-line therapy. I should also mention that there are still also regions in the world where lorlatinib, unfortunately, is not available or reimbursed. And so in those countries, those patients will at least have potentially access to NVL 655 or alectinib. So does that answer most of the questions?
I think you covered many of them in the chat. We're not going to get to them all, but I think that's a really good one. One of the other questions that comes up in here and is pertinent perhaps to most people on the call is, what do you do what do you do when you progress on first line lorlatinib? You gave us these cool orthogonal approaches, but that's not going to be possible for many. So what is sort of your sequence now for how you treat patients who start on alectinib or start on lorlatinib when they progress?
Yeah, so there's not that, I mean, the experience we have with first-line lorlatinib still is almost all from the CROWN study. And like I said, because the uptake was only pretty much starting after the CROWN 5-year data was presented in June. So, and of course, if we're now starting to prescribe patients first-line lorlatinib in the last 6 months, I can say they're almost all doing great. So we don't have as much experience with patients relapsing on first-line lorlatinib yet. And in fact, I have been talking with Pfizer. Of course, they're still following those patients who are on the CROWN study. And there's quite limited data to understand why some of the patients in the CROWN study relapsed on first-line lorlatinib. We presented in the 5-year update that in the analysis of liquid biopsies that were done at the time when patients progressed on first-line lorlatinib, no ALK resistance mutations were identified in about 30 or so patients. So we don't yet know, Ken, much about what will drive resistance to first-line lorlatinib. It certainly should not be a single ALK resistance mutation. Could it be a compound ALK resistance mutation in some patients, perhaps? But if I had to guess, it's going to be something completely not ALK related. It's going to be not an ALK independent mechanism of resistance. So I would say the jury's still out on what drives resistance to first-line lorlatinib. And because of that, I would say we don't yet have the therapeutic strategies on hand to then offer patients who may develop resistance to first-line lorlatinib. But this is a really important topic. And so I do think that hopefully we can all put our heads together and think about how we learn more about these mechanisms of resistance to first-line lorlatinib. Now, alectinib is a very different situation. We know a lot about resistance to alectinib. Alectinib given second line, third line, and also alectinib given first line. And we know that in about 50% of cases, when patients develop resistance to alectinib, they've acquired a new secondary resistance mutation within ALK. And that's now causing alectinib to no longer be able to bind and inhibit ALK. And so those cancers tend to still be very ALK dependent or ALK driven. And hence, more potent drugs that can overcome those resistance mutations like lorlatinib, like NVL 655 can be really, really effective in those cases. I showed you the data with NVL 655 that again, NVL 655 has activity across the board, but where we see especially good activity is exactly in those cases where there are ALK resistance mutations and the cancers are still really dependent on ALK. I would say the most important thing to do if you're on alectinib or brigatinib or any of these TKIs, and there's some signs of resistance developing, is a biopsy is really important. You can start with a liquid biopsy, but many of you know that liquid biopsies do, have some limitations in terms of sensitivity. And so I think if the liquid biopsy is uninformative, then a tumor biopsy, I think, is warranted to try to identify the mechanism of resistance and then to be able to tailor the next line of therapy based on that information.
So Alice, in your experience, how often is it that you can't technically get a biopsy from someone? Because you've hit on a really important point, right? Everybody should get a biopsy if they think there's progression, but there's a lot of people who don't get them. What should that expectation be?
Well, certainly liquid biopsies are the easiest can, so that's nearly, accessibility, access is one thing, but a liquid biopsy technically can be done, right? It's typically just a blood draw. But you're right, tumor biopsies are not always feasible. I think tumor biopsies, in my experience in patients who are relapsing, probably about maybe even 50% of cases, a biopsy could be feasible. But then probably half of those patients may opt to not proceed with a biopsy. Biopsies are, they are an invasive procedure. They do come with some risks and inconvenience. And so if the patients sometimes will opt not to. And in that case, then we can still proceed with the next line of treatment, but then we aren't able to really tailor that based on the underlying mechanism of resistance. But I would say, compared to when I think the liquid biopsies have been amazing. They've really allowed us to have so much more insight into the biology of resistance. On the other hand, they do not completely replace tumor biopsies, right? Because of the limitations that I mentioned. Oftentimes a tumor biopsy is really important to get.
So I'm going to ask one more question, and then Mark and Summer, I'm going to pass it back to you, because there are a lot of very sort of case-specific questions in the chat, and I thought you guys might want to pick out the ones that you thought were most appropriate. I'd like to finish to talk a little bit about MTAP, because one of the questions in the chat was, how do you know if you're MTAP deleted?
Yes.
Well, just for the group to know, if you look in your NGS report, people are sending me, who are a part of our committees, their NGS reports, and there's a lot of information in there, including MTAP deletion. Now, I don't know how long those have been included in the Foundation Medicine, Caris, and other reports, but there's a lot of information in those reports that are really valuable that your physician should be aware of and should be taking into consideration for your treatment planning. So I wondered, some of the questions around MTAP are, how long has it been in the NGS report? Mm-hmm. And do any of these PRMT inhibitors cross the blood-brain barrier, for example? Maybe that's part of what you're planning to do. And I'm kind of curious, do you know of any of these companies who are doing clinical trials with their PRMT inhibitors treated outpatients besides yours? In other words, is there any additional information that could give us confidence that to help us expedite the study of this with a TKI in our patient population?
Yeah, so all really, really great questions. So PRMT5 is, I mentioned it's a synthetic lethal target in the setting of MTAP loss. That was discovered in about 2016 timeframe. And then PRMT5 inhibitors, it's a long story in terms of how these drugs have come to the clinic, but they have been in the clinic for some time. But I think that demonstration in 2016 really, made the folks who do the sequencing realize that they've got to be calling MTAP on these NGS reports. I was just chatting with another ALK patient just a few hours ago, and she shared a Foundation Medicine report with me from 2017, Ken. And at that time, they were not yet looking for MTAP. I even asked Foundation Medicine, I had reached out to them to say, well, can you just go back and look at the sequencing reads and look for MTAP? And they couldn't, they weren't set up at all to look at MTAP. So I can't tell you for every assay when they actually now started looking for MTAP, but I can tell you Foundation Medicine wasn't doing it in 2017. It probably was definitely more in the last 5 years or so that some of the NGS companies have been looking for MTAP. I should also mention to the earlier point around biopsies, liquid versus tumor biopsies. So I mentioned that MTAP is a tumor suppressor. You need to have loss of both copies for this to be a relevant target. And it's very hard, if not impossible, to detect MTAP loss in a liquid biopsy. In a liquid biopsy, as you all may know, we're trying to detect a very small amount of circulating tumor DNA in the bloodstream, and that's in the midst of a ton of other normal DNA from your normal cells. And the normal cells, of course, have MTAP. And so it's very hard to detect true loss of MTAP in a liquid biopsy. You really need a tumor biopsy. And even the tumor biopsy technically can be challenging for other reasons that I won't go into. But tumor biopsy is necessary for detecting MTAP loss. It doesn't have to be next generation sequencing. It can be immunohistochemistry, which some of you know can be done off of one single slide that can be done to look for expression or no expression of MTAP. So this is an important target. It's not all ALK patients, right? It's 30%, but 30% is, in my mind, is still a significant fraction of patients who could have another targeted therapy option. The other question you asked, Ken, is really, really good around CNS penetration of this new class of drugs. So these PRMT5 inhibitors. So several of them have no CNS penetration, which I think is going to be a liability, not just for ALK, but for other lung cancer patients, other patients. But now there are several that have entered the clinic that do have CNS penetration, and some that are even designed specifically to have CNS penetration. And so sort of in my ideal world, when I think about what would be the best investigator-initiated trial when we're looking to test this concept of ALK plus PRMT5 inhibitors, we need the best ALK inhibitor, our best anchor, and we need our best CNS penetrant PRMT5 inhibitor. And so, Ken, they do exist, but they're all in early phase clinical trials.
Alice, Mark, Summer, where would you like to go next?
Yeah, I want to pivot back to NVL with the phase 3. In the phase 3, are they going to reduce down the amount of uh, disease that you need to get into the trial, or is it going to be the same requirement of 1 centimeters
No, it's
of
still the
measurable
same. It's
disease?
still the same requirement for measurable disease.
And I want people to understand that you just can't flip into the trial without an active disease.
Yeah, and remember, Mark, for the Nuvalent phase, are you talking about the phase 2 that's ongoing
No,
or the
I'm
phase
talking
3?
about going into phase 3.
Yeah, so the phase 3 is for newly diagnosed patients. So
Mhm.
I think most of those patients will likely have measurable disease, but that is a requirement.
Right. So you just
And
can't flip into that drug. People have to understand that. You have to have active disease going on,
Well, and you
progression.
also can't have received a prior ALK inhibitor, right? So you're
Right.
new.
Well, that's for the phase 3 is what you're saying. Phase 2 still will be open.
Yes, so phase 2 right now is enrolling. Basically, again, the Nuvalent team has been wonderful, and they have those 6 expansion cohorts that I showed that are enrolling basically ALK patients with almost any history of prior treatments, TKIs, chemotherapy, any prior treatments. They've enrolled rapidly and significantly, as you can imagine, right? There's a large need here. So there's just a lot of experience we now have with this drug. And at some point, I hope in the near future, they will flip into an expanded access program for NVL 655, which may make it more accessible
Mhm.
and a lot more easier for our patients to receive this drug.
Since we have a lot of newly diagnosed people in the last 2 years there, when do you get a second opinion?
Ah,
What's your suggestion?
yeah. So I mean, my personal opinion is that second opinions can only help. And I would say, especially, well, first of all, I think for anyone who's faced with a serious disease, and I would say a diagnosis of lung cancer is serious. And then I would say also for someone who has a relatively rare type of lung cancer, which ALK is, I think you all know that you are pretty rare. Only 5% of our patients have an ALK rearrangement. I think that also is a reason to pursue a second opinion and be seen in the center where there's an oncologist and a team who has a lot of experience treating patients with ALK positive lung cancer because you guys are different than other lung cancer patients, and the treatments, and you can probably appreciate this, are a lot more complicated these days. In the old days, 10 years ago, as I showed you, we just had crizotinib and then maybe one other ALK inhibitor. It's a lot more complicated now with, 4 generations of ALK inhibitors. We often add chemo to an ALK inhibitor. We have clinical trials of combinations. We do all sorts of things. We go after local disease, right? If there's 2 little nodules that are growing up, we're gonna try and radiate those or remove those, right? So we do things quite differently for ALK patients compared to non-ALK patients. And my worry is that if you're at a local center where perhaps your oncologist has either never seen an ALK patient or only has one ALK patient a year, they're just not going to have that same level of experience. And so I do think it's often helpful to have a second opinion. And then often that can be a resource for your oncologist, someone who your oncologist can continue to reach out to over the next number of years as you're on your different treatments.
That was a great point. Thank you so much. And I think that people sometimes are scared to get the second opinion. They're comfortable where they are, but they can still go back and use a second opinion as a resource. Exactly. This has been a wealth of information. I want to go back to Nuvalent again, please, and just clarify that phase 2 will stay open as phase 3 launches, correct?
So I don't have all the details around what they plan to do with the phase 2 once the phase 3 opens, but I mentioned the expanded access program.
Correct.
So there will be an expanded access program that will open so that there will hopefully be minimal gap between when the phase 2 does ultimately close
Right.
and then how patients access. Because remember, the phase 3 is only for newly diagnosed patients.
Exactly.
And then if the phase 2 were to close, that means anyone other than newly diagnosed patients would not be able to access it. So their plan is to have this EAP, expanded access program,
Yeah.
coming very soon at all the sites that have the ongoing phase 2, so that there's no significant sort of gap in time so that patients, can't access NVL 655.
Okay, perfect. That's exactly why I just wanted to reiterate that. So there's not a panic. There were questions about, and you actually laid out the process that when you experience progression, we all know that that is hovering out there at some point, that then the importance of a biopsy. And then from there, somebody was saying, okay, well, if they're on alectinib, typically then they would biopsy if it's still ALK driven or go on to lorlatinib in the past. Well, now Nuvalent is an option. So or do you feel like you're divvying them up more?
Yeah, it's a great question. One of the expansions, or one of the cohorts within the phase 1 and now the phase 2 has a focus on patients who had received prior ALK inhibitors, but not lorlatinib. And in fact, those patients did really well. I didn't emphasize that on the slide because there was just too much information, but the patients, for example, who had been on first-line alectinib and then went on to NVL 655 did really well. I think the response rate was above 50%, and the durations that are emerging are really, really promising. So it looks like, again, in cross-trial comparisons, that NVL 655 looks at least as comparable as lorlatinib in terms of potency, in terms of CNS effects, but comes with many fewer side effects. And so when I have a patient who I'm meeting or I'm following and they are showing signs of progression on alectinib, which we cannot treat with just local therapy, for example, and we know we need a change in systemic therapy, we are talking based on their biopsy samples about next steps. And if it's appropriate for another ALK inhibitor, I am talking about both lorlatinib as a standard option, and then also I'm offering Nuvalent 655 on the clinical trial for them as well.
Fabulous. And also, there were some questions about the TRK pathway, what exactly that was, and what side effects come with that.
Yes, thank you. So TRK is a protein it's a receptor tyrosine kinase. It's related to ALK, and it is often inhibited by drugs that target ALK, drugs that target ROS. And in fact, TRK can be a driver of cancer itself. There are TRK rearranged lung cancers as well in about 0.1% of our patients. The issue with TRK is that it is expressed on some normal cells, particularly neurons and other normal cells. And so if you inhibit TRK in the brain, for example. We do think that that does likely cause some side effects such as dizziness, sometimes being off balance. It may mediate increased appetite and weight gain that we see with lorlatinib. And there is even a suggestion that inhibiting TRK could cause or contribute to some of the neurocognitive side effects that I talked about with lorlatinib. And so again, the Nuvalent team was wonderful. They went into this wanting to make the best ALK inhibitor possible. And, we had talked about these side effects that were really somewhat challenging in some of our patients on lorlatinib. And so what they did was they made an even more selective ALK inhibitor that would not inhibit TRK, trying to basically remove this liability related to inhibition of this other protein. And I think they've done a great job because in that safety profile I showed you, there isn't any dizziness, there isn't weight gain, there aren't any neurocognitive or mood side effects that have really been noted with NVL-655. So I do think that dialing out this TRK activity has been really, really helpful for NVL-655.
For sure. We all cheered for that. How about a hot topic in our support groups often are GLP-1s or glucose inhibitors, I guess you would say, such as Manjaro or Worvoy. What do you have to say about that? And do you think that we're good candidates?
Yes. So I had a few patients, and actually there's even at least one published case report on an ALK patient who went onto one of these GLP-1 agonists. I don't remember now which one it was, but we have seen some really nice results with these for patients who are on alectinib and have had some issues with weight gain, as well as patients who've been on lorlatinib where the weight gain is even more pronounced. So at least in the published case report, this patient did derive a lot of benefit in terms of weight loss, tolerated that GLP-1 agonist as well. I think it's a reasonable option if you've exhausted the typical options that we recommend to manage sort of the increased appetite and weight gain. So my first sort of advice usually even before we start on alectinib or lorlatinib is that watching what you eat and also exercise activity is really, really important. And for patients who are really disciplined and can be careful about what they're eating, recognize those sort of hunger signals and not go for the carbohydrates, but go for the sort of the low carbohydrate snacks. They can really manage oftentimes the weight gain really well. So we always try sort of dietary behavior modifications first. But I have had patients who've done a really great job with all of that and they still gain weight. And so in that case, I am open to considering GLP-1 agonists, again, because we have at least one published case report as well as a number of anecdotal reports of patients doing well.
That is awesome. We always like to have more tools. Mark, do you have
I got
anything?
2 minutes. I'll do a lightning round.
Okay.
So,
And I saw some vaccine questions too.
yeah, we had a ton of questions about all the great things that you just shared with us. Do you do blood test markers?
So I think you're referring to markers like CEA.
Yeah.
I don't routinely do those, although quite a few patients who come to see me are already having their CEAs tracked by their local oncologists. And I think if they feel strongly about it, or if I look and I see that their CEA is actually tracking reasonably well, I will continue that. But I don't typically start off tracking patients. We have some experience now. This is like, I don't know, in the early days of the phase 1 LDK 378 trial, Ken will appreciate this. We actually did collect CEA values for patients on the phase 1 study of this ceritinib or second generation ALK inhibitor. And in some patients, it was a minority, it did track with their disease, but in the majority of patients, it didn't. And you can imagine then an elevation in CEA could very well be misleading, kind of be a false positive in a way and lead patients down a very stressful path in terms of further workup. So I don't routinely do it for all patients, but I will continue it if it's been started and tracks well with the disease.
Okay. Scan schedules and when do you do PET scans?
That's a great question too. So a lot of our patients, especially if they've been on their TKI, say for the first, they've just started their TKI, we'll be getting scans typically pretty early to make sure we're headed the right way. But then we'll usually get into a schedule of about every 3 months for what I do is CT scans every 3 months for the first, I would say, couple of years. And if they have a reason for a PET scan based on the CAT scan, then of course we will get the PET scan. But I don't routinely screen patients with PET scans, and definitely not every 3 months. That's just like a lot of radiation. Now, there are just a few exceptions here, though, Mark. If a patient has, for example, a disease that's really difficult to see on CT, bone disease, for example, sometimes liver disease is hard to see. And especially if a patient has a contrast allergy, which makes it even harder to see things, especially like in the liver, there may be reasons to get a PET scan. But even then, I tend to alternate CT and PET just because getting a PET scan every 3 months is just like a lot of radiation.
Last question. Will you come back?
Of course, I'd love to come back.
All right. And you left us with so much information I did not expect at all. So, and we
okay.
have a ton of questions in regards to your slides. So we will have to have you come back and update us with
Absolutely. I'd
Ken
be
and,
happy
and our
to.
medical communities of what's going on. So thank you so much. And this
course.
is and I want to stress to people that, I mean, you are giving up your Sunday, 1 hour in your Sunday to come and join us. And without yourself and other professionals and doctors, researchers, it wouldn't happen. And you were one of our first guests back in 2021. So thank you again.
Of course. Thank you.
Excuse me, can I say something? Yeah,
It's unusual, but you can say one thing really quickly.
okay. Yeah. First, I want to thank Dr. Alice, because I'm really blessed through Mass General Hospital Lung Cancer Center. My doctor respects you very much, and I have been on the clinical trials for about 3.5 years. Yeah,
Great.
which is the ALKtALK plus metinib.
Excellent.
Yeah. So after two years, I become complete responder to this trial. Yeah, phase 1/2.
Amazing. We started that many years ago, so that's amazing to hear. Thank you for sharing that.
Yeah. So at first, you had to lead the cancer center, lung cancer so well. So I'm, had no chance in your treatment, but, the whole center just give our patient much, much trust and, encouragement. Yeah.
Great.
So can you just quickly comment about this combination with this cobimetinib?
Yeah, so that was a combination we started based on a number of different preclinical studies, including one from Trevor Bavona's group in UCSF, that really suggested that combining an ALK inhibitor with cobimetinib, which is a MEK inhibitor, so it targets downstream of ALK in the MAP kinase pathway, could be particularly effective, especially in preventing resistance from emerging. So he did some beautiful preclinical studies. And then in our lab at Mass General, we also had data to support co-targeting ALK and this MEK protein to again suggest that the duration of response would be even better than with ALK alone. And that's what led to the phase 1 trial. And we have had a number of patients do quite well on that study. It's great to hear and see you. But we've also had patients who have struggled a bit with some of the side effects. And I would say that combination therapies are hard for patients. They're hard to develop as well. But I think we need to figure out and do more around combinations. I do think I've kind of interweaved that into my talk tonight, that we need to go beyond just hitting ALK itself. We have to go beyond and think about other important targets within ALK cancer cells. Cobimetinib may be targeting an important one. It's a little bit, I would say the toxicities can be hard for some patients, but I think there are also other targets. I mentioned one tonight with MTAP that we should also explore in combination. So there's a lot of work to do, and I think these combinations are the way to go for the future.
Thank you so much. And you guys can unmute and say thank you, and we'll see you another time.
Thank you, everyone.
Thank
Thank
you.
Dr. Shaw.
Thank you, Dr. Shaw.
Thank you very
Thank
much.
you.
Right.
Thank
Dr.
you.
Shaw, have a great day.
Alice.
Thank you.
Thank you.
Thank you very much, Dr. Shaw.
Thank you.
Take care, everyone.
Thank you, Dr. Shaw.
Thank you.